Good afternoon and welcome to Energene's webinar series. My name is Yana and I'll be your moderator for today. We'll wait a couple minutes to let everyone to connect and start shortly. Okay, I see that people are connecting and I believe we can begin. Today we'll be discussing the advantages and the logistics of working with both clonally propagated and seeded varieties, and how applying genomics accelerates their improvement. We will save time for questions at the end of the webinar, and we encourage you to type in your questions during the presentation using the Questions tab on the left of your screen. This webinar is also recorded and will be posted on our website in case you want to listen to it once again. It's my pleasure to present today's speakers. With us today, Yannik Schlup. He's one of the founders and Chief Business Officer of Puregene Ag, one of our most advanced customers. Puregene is a state-of-art cannabis and hemp breeding company. Yannik has an extensive background in business development and molecular breeding, and we are very excited that he was able to join us today. Those of you who are regularly attend our webinars are already familiar with our next speaker, Dr. Uri Weishaus. Uri is with Energene since 2011, applying genomic tools for marker development and breeding projects of various crops. Lately, he has specialized in cannabis genetics and became an expert in the field. Uri holds a PhD in plant and microbial genetics from the Hebrew University, and his academic background is in genetics of plant secondary metabolism pathways. Uri, the stage is yours.
Thank you, Yana, for the introduction. So I'll start with, before we move to our guest today, I'll start with the short introduction on terms. We know that there's very diverse audience coming from the business side and from the academic science and sales side. So we try to keep it fit for everybody. So first of all, I'll explain the difference between cannabis, marijuana, and hemp, because they're interchangeable when people talk about breeding, but there are some distinctions. So the plant species is cannabis, and it divides into two major groups. One is called hemp. They're all crossable. It's the same plant, but humans have made a distinction between hemp and marijuana type, or broadly used cannabis, which is usually used for THC extraction in various forms of the plant, flower or extracts, with THC content above 0.3%, but usually in drug type varieties, there's more than 10% THC, whereas the other types of cannabis are called hemp, and they're used for various things: fiber, grain, protein, oils, and CBD. And legally, there's a strong definition, a threshold of 0.3%. Beyond that, it's cannabis. Below that, it's hemp. And this definition, as Yannik will explain later, changes with geography. Within the group of hemp, there's not clear cut, but people refer to two kinds of hemp. One is industrial hemp, and one is CBD hemp. So industrial hemps are usually used for the uses that are not CBD, meaning fiber, grain, oils, biomass, etc., and usually originate from classic varieties coming from mostly Northern Europe. And then there's CBD hemp, which was developed with various genetic backgrounds used for CBD production. They usually contain above 3% CBD, most of the times more than 5% CBD, and in most or many of the cases, they also produce THC when fully ripe. So a major distinction when talking about breeding for varieties with seeds, we have to understand that there's a difference between a heterozygous plant and an inbred or a fully homozygous plant. So cannabis prefers outcrossing, meaning it prefers not to be homozygous. Most of the genome is heterozygous, meaning you have two copies that may be different in each locus, depicted here in different colors, say here gray and orange. This is an example of two sets or a set of two chromosomes, two copies of the chromosome. Whereas a homozygous would be identical throughout the chromosomes. So in what people use most of in most of the industry nowadays are seeds made from a cross of two varieties. They're two heterozygous or mostly heterozygous varieties, and they create a set of seeds. These seeds, people may refer to them as F1, but they're not true F1 seeds, but they're hybrid seeds. And because they come from parents with heterozygous backgrounds, or mostly heterozygous backgrounds, they may segregate in the field. And that's why you see when you buy seeds from not so reliable sources, you may see segregation of traits in the field. Plants are taller or shorter, have different flowering times and so on. And the performance of the varieties or plants is maybe poor. Whereas true hybrid seeds, you take two inbred lines, and as we said, there's high inbreeding depression, meaning in advanced generations of selfing, the plants cease to produce viable seeds or make very low number of viable seeds. So the cannabis plant does not prefer to be fully inbred. So what we do is have two inbred-like parents or nearly inbred parents, meaning most of the genome is stabilized. And then there are some regions that are not associated with major traits that may be heterozygous. And when you make a cross like this, all of the seeds are very close to what other crops call true F1 seeds. They're nearly identical. There's one copy from each parent. And for all practical purposes, they perform as identical F1 seeds in the field. And this is what we aim to do when we're trying to make hybrid seeds that are stable and perform well in the field. Another type of propagation, usually associated with marijuana type or drug type indoor grows, but also for CBD grows. People use clones, meaning you have a mother stalk, you make cuttings out of it. It's fully heterozygous, whatever the gene content or the genome content of the plant is identical to the cuttings. They all share the same genetic makeup. Usually this is done in hemp and CBD hemp and indoor grow, whereas the other types of grow are characteristics of field grow, row crops. So that's more suitable for hemp types, but I've also seen both forms in CBD and hemp. One major distinction is with true hybrid seeds, most of the seeds would be feminized seeds and you would have no males in the population, whereas in common seeds, which are crosses between two nearly heterozygous varieties, you would most of the times have males in the population that you either cull out or maintain in the grow because they do not disturb your purposes. With that, I will end the introduction part and the terminology, and I'll give the stage to Yannik. Yannik, it's a pleasure to have you with us today to talk about Puregene and what you're doing with genomics. Yannik, yours.
Thank you to you, Uri and Diana, for the introduction, and thank you to Energene for organizing this webinar. It's a pleasure to be invited as a speaker. With our company, Puregene, we've been on the cannabis market for almost 3 years. Operating from Switzerland, we're based in one of the most active cannabis hubs worldwide. We've gained lots of experience from which I'd like to share some valuable insights with you today, and I'm very happy to do so. First, I'll present to you briefly what Puregene does, then I'll go over the major markets and their differences, meaning Switzerland, Europe, and the US market mostly. Then I will walk you through genomic breeding and lastly exemplify how those tools, or let's call them breeding assistants, accelerate and improve cannabis breeding, whether it is seeds or clones based. I hope in the time of coronavirus, all participants are safe and healthy. And let's start with the presentation. A brief history of Puregene. It is part of the Pure Holding Group, which is a group that started cultivating low THC cannabis or low THC hemp, meaning it's the cannabis plant cultivated for flower, almost 4 years ago. And it began with cultivation only, and then over the course of time, the group specialized into various fields across the value chain, and in the meantime spans everything from varieties breeding and development all the way overproduction, all the way to B2B, B2C operations and pharmaceuticals. So within this framework, Puregene is the company that provides genetics for the holding and develops genetics overall for the world hemp market. Puregene, in its very activities, is essentially a breeding company that breeds and develops state-of-the-art varieties, meeting the requirements of the agriculture industry, so the modern agriculture industry. Like Ori has briefly mentioned, hemp is becoming more and more, say, commonplace again, luckily, and therefore the requirements increase. The plant needs to meet standards of row crops, it needs to be stable. And this is one of the specializations of Puregene. Additionally, we have positioned ourselves and are here to help breeders tailor or optimize their own varieties. We have heavily invested in genomics, since this is the modern way of breeding plants. I'll exemplify this later, and Ori will then give a beautiful overview over what's possible with Energene. I've been working with Energene for quite a few years now. And there also we had started with the pangenomic project. I'll get into details on what a pangenome is too. It's, in brief, a high quality reference genome used for molecular breeding. The cannabis market has its intricacies. It's not only that it requires breeding, it's also the regulation as an additional layer of complexity. And currently, to us being active in 3 markets, Switzerland, the European Union, and the US, there is, I'd say, 3 types of markets, which I would like to briefly walk you through. In Switzerland, our home place, the regulations are as follows. It allows up to 1% total THC at harvest. To briefly get into the THC, I'd even call it debate. There's 2 types of molecules. There's the acidic form, THCA, and delta-9 THC. And depending on the regulation and the interpretation thereof, the total THC amount is either the sum or only delta-9. In Switzerland, what's relevant is the sum of the THC, and it applies to all organs of the plant, meaning a trim flower must have below 1% THC, so does a leaf, but whatever product comes out of cultivation has to stay below 1%. Then in Europe, it's much stricter. There is a limit imposed of 0.2% total THC, meaning 5 times less, breeding becomes very complicated with such regulations. And additionally, in the European market and Switzerland also, compared to the United States now, there's an additional regulatory hurdle, that is the variety registration, meaning a variety needs to go to trialing processes and registrations, which takes up to 4 years before it can be sold. In the US, it's different. Usually registration is either not necessary or much faster. And there's this 0.3% THC limit. Especially with the law changes and reinterpretations recently, there's plenty of room for interpretation and therefore also uncertainty. It is a debate whether it's total THC or only delta-9 THC. And what it shows is how important it is to have a quick acting and reactive breeding program that within a short period of time can account for such changes or always be ready for additional uncertainty. As I briefly mentioned, Puregene started in collaboration with Energene the Pangenomic project a few years ago. And the reason why I'd like to present the pangenome to you briefly is because breeding always requires a reference genome. And a reference genome is the orientation frame for a molecular breeding program. In the next slide, I'll walk you through molecular breeding, and after you'll see Energene's offer, which is also based on high-quality reference genomes. A reference genome is a phased genome that distinguishes the heterozygous phases of cannabis. Unlike many other row crops, cannabis is highly heterozygous, which is an additional challenge. The pangenome that came out of this collaboration is one of the highest quality genomes and can be compared to the Human Genome Project. And with it, it captures the variation between different varieties. Hemp is with lots of origins. It's a debate where it exactly comes from, but it's also a fact that depending on which hemp varieties you look at, genetically speaking, there is lots of variation between, say, a Russian origin, European, and African origin. And this is all captured in pangenome and then allows to differentiate between variations, and this is the starting point for a breeding program. You need variation with which you can play to get to new varieties. Starting from a reference genome, there comes the molecular breeding of clones and seeds. Clones and seeds breeding differ. You could say that seeds breeding is a sequel to clones breeding, but I'll get into more detail after. All in all, molecular breeding needs full commitment. It is there, especially in hemp, to bridge the gap to breed modern hemp varieties, because hemp hasn't been bred in a way, say, soy or maize has been bred over the last decades, and there's this gap to bridge. And phenohunting, meaning classic selection, won't be sufficient to meet the pace of the industry and close this gap. Therefore, molecular breeding is what offers the higher precision and faster pace to market. And with high throughput, all the marker-assisted breeding, the genomic selection, new biochemistries can be unlocked. Better varieties with, say, multi-loci and complex traits can be bred, and in a much more targeted way than without the molecular support, the molecular assistant in a breeding program. There's this important notion of molecular breeding. It's a full-term commitment, and Energene will present a very, say, supportive way of going about it. If one does it themselves, it requires experts from the fields of biochemistry, of molecular breeding, etc., etc. And here I would like to present to you a brief case study of how this can work. For instance, we hold varieties with an extremely high variance proportion. And now, if a cannabis breeder or company would like to have high variance varieties, such as, say, a CBDV, they bring in their varieties that has good yield, quality traits, and say a lovely flavor bouquet. And then, thanks to the genomic knowledge, mostly the pangenomic knowledge, their variety can be IP protected, and at the same time, extremely quickly, the variance trait, for instance, THCV, can be introgressed into the client's variety, and then this variety is available. For clones, as you can now tell, there's the difference between clones and seeds. This is a fast process. If you understand the markers and the inheritance of genes or complex traits, those can be bred in few generations, and within less than a year, you get to this tailored variety. However, for a seed variety, then the complexity increases since it requires more stability. And like Ori's mentioned, cannabis has its intricacies in terms of stability, since it can't be fully introgressed for, apologies, it can't be fully self for many generations due to the deleterious alleles. And then with the genetic tools, with marker-assisted backcrosses, from a clonal variety in another few months, meaning 1.5 years to 2 years, stabilized seed varieties can be bred. So the difference between clones and seeds, apart from many other things I'll get to after, is especially the complexity in the breeding process. To present to you some of our experiences in the clone market, on the right you see one of our fields we had cultivated in the group, which was an outdoor crop of a clonal variety. It is faster to breed, and it's exact replicas of the parental genetics. Traits can be introgressed fairly easily, since it just requires this one seed that has the right combination. This seed is grown up and mass propagated. However, the propagation is much more expensive. The going to market is also not that simple. And moreover, a big downside is it's presumably genetic drift within clones. Their vitality just decreases over the cycles of generation of propagation. There is probably lots of you who have observed that if you have a clonal line 2 years later, the performance has decreased. And this is one of the big downsides of clonal lines. And then there's the seed market. There is this one bigger challenge of feminized seed, since if it's male and female plants, what you will get out of your crop is seeds instead of non-pollinated flowers, which then produce, for instance, all the CBD or CBG the cultivator is looking for. So this process of seed feminization is extremely relevant to master, since otherwise a clonal line who's solely female definitely performs much better than a seeded line. The upsides are that it's very easy to be propagated and the price point lies much lower. However, in terms of the breeding schemes, like Ori has briefly touched upon, it's a much larger challenge, And especially in a highly heterozygous crop like hemp, it's a major hurdle to fully stabilize the genetic. And lastly, since stabilization always has to go hand in hand with traits introgression, this leads to a delay, which in an industry like the fast-paced cannabis and hemp industry, when 1 month is probably like a full year, it can be a challenge and requires lots of predictions and flexibility. To give a few last examples over what such a molecular breeding program can lead to is some of the varieties. Without going into too much detail, there's different cannabinoids such as CBD and CBG, and then also the variants such as chemotype dominant CBDV varieties, CBGV varieties, THCV varieties. But what's much more important to me to show to you here is that through the whole molecular breeding, the background, the platforms, it is possible to, within a few years, breed dominant chemotypes, novel traits, even very complex traits that are put to or dictated by many genes, and then stabilize those plants into seed lines. So that was it as a brief overview over the markets, the clones versus seed debate. And I would like to hand over now again to Uri, and he will give you more insights over the offer from Energene.
Thank you, Yannik. This was super interesting and very informative on the European markets, and also on actually applying genomic tools in the breeding process. One of the things that Energene does for its clients is a end-to-end solution of the bioinformatics part. Anything that is outside of growing, which is the one thing that we do not do, you send us the DNA and we take it from there and deliver back, in a nutshell, the breeding decisions. We help you design and implement the actual, define the breeding targets, define the right populations, analyze the populations, and find, generate markers that help reach the targets very efficiently and much faster. We have a general package called Quick Genetics, which pretty much includes everything: the diversity analysis, consultation hours, exploring your genetic makeup, analyzing and building your own reference genomes, and exploring specific populations and mapping specific and complex traits that are chosen by the clients. So the first step would be the diversity analysis or broad diversity analysis. Usually people have vast collections of somewhat characterized or fully characterized varieties, either as seeds or as clones. Sometimes the clients know a lot about their collections. Sometimes they need to manufacture data. Usually these are a lot of plants that need to be culled, and the number that we have to work with usually is much smaller than the number in the collection. So what we have to do first is analyze the collection, correlate it with phenotype data as much as possible. If not with phenotype data, then we just need use genetic data, make phylogenetic trees, and determine genetic distances. And then we could select individual founders based on a combination of genetic distance and phenotypic performance. So it's the most efficient way to actually reduce the number of plants and create founder lines. So the next step would be to select individuals that are farther away genetically from one another and incorporate them in breeding programs as founder lines, meaning usually we have one common parent. Usually that's the variety that the client likes to use best, its moneymaker or the backbone of its breeding program, something that you already know very well and grow. So usually that would be one to fully analyze as one of the parents, and with that we could make 2, 3, or more populations, either as true crossed with a true male or with a feminized plant, or made or female that's been reversed to provide pollen. And then we create populations of about 200 plants each, and we analyze the set of populations for the specific traits that the clients request. So in a deeper overview here, and this is an example for flowering time. This also has to be provided by the clients because we're not growing, we don't have a greenhouse. You have to provide us as clients the phenotype data, the traits that you're after, in this case flowering times. And then we would take DNA from both parents. They have to be extremes. So you would want to cross varieties that flower early and varieties that flower late. We take in 2 heterozygous, fully heterozygous clones or varieties. We don't have to have inbred lines, which is very unique in the process of breeding. We generate a segregating populations for the trait, and the quick genetics package incorporates 5 traits of choice. In this case, it would be flowering time, and then you have 4 other traits in the same population to actually map. And we would map and isolate the specific regions in each of the parents, right? Each one of these colors here, dark blue, light blue, yellowish or greenish, we determine the exact phase that has to be associated with their phenotype of choice. So for instance, you want shorter flowering time, you have to have the dark green version of the genome. And we generate markers that are specific to the dark green version. Or if it's a combination of both, then we know and generate markers for the right combination of both. Specifically, once we've found a region of interest, we could zoom in and start exploring the region of interest for specific genes and generate new IP, whether it's gene regulatory sequences or actual candidates for research, further research on their function and whether they could be edited and so on. So this is sort of a secondary step, whereas you first of all find markers and don't even care what they do. You just have a means to select for what you want. And then the second step would be to actually zoom in and study what you've found, and it's all novel IP belonging to the client. So, sorry about this. So in a general overview, breeding is usually divided nowadays into 2. There's conventional breeding, which is what most people prefer and do. And then there is genome editing, which is a novel form of changing the genetic content or artificial interference. It's regulatorily more complicated, but there is a lot of interest in this new technology. So I'll elaborate on what we have for this. But for classic breeding, when you want to introduce a trait, whatever the trait is, you have your elite variety and you have a donor. So with classic breeding with molecular markers, you need to integrate the trait of choice or multiple traits of choice back into your elite variety. For instance, have a good performing variety that flowers earlier. You would need to do the cross and then use the molecular markers found by us to do multiple cycles of backcross till you have the exact elite variety with the desirable traits you want. But then again, a lot of the clients are not interested in this because it takes longer. So they do want to go towards the genome editing space, whether to generate actual plants or to generate synthetic biology and discard the plants. So with genome editing, CRISPR-Cas9 or other versions and other tools for editing, you have to first identify your target. You need to find out what to edit and what you want to edit to. You want to knock out a gene, you want to duplicate it, you want to overexpress it, you want to modify it. all of this has to come from analyzing and interrogating the actual germplasm that you have and associating functionality with the new gene or the new genetic content. Once you have the edit, usually you need to do back crosses in order to reach homozygosity or to have multiple copies or so on of the edit, or at least eliminate some of the edits. So you also need markers and more information about your genome to have good editing capability. So for that, we have delay in slide switching. For that, we've come up with Energene. So Energene is a vast curated cannabis database that contains unique proprietary genomes that we've sequenced and assembled over 14 genomes currently, and pretty soon we're going to have lots more. All of them assembled by Energene and compared all to all. And we've also compared it to publicly released genomes in the public domain. So what this allows is for companies either not interested at all in the plant, but only in the genetic content, or interested in the plant, but cannot have in-house genomic capabilities. So for that, we have the Energene, which allows you to compare multiple genomes to drill down into regions of interest, either QTL mapping, through homology-based searches, or any other ways of regions of interest. And you can look at multiple genomes in a specific regions. There's a very strong visualization tool that allows comparison of multiple genomes. And the main features that you can do with this is look at regulatory elements, look at present absence variations, true present absence variation within the genomes or different phases of the genomes, because we phase our genome assemblies. We could look at true copy number variation, and we could also discover new alleles and novel genes. We've already done this and discovered at least 12 novel alleles for the THCA or CBDA synthase in the assemblies we've made. So as an example, if you're into synthetic biology and your company does not want to breed plants, but wants to take genes from cannabis plant, we've analyzed the content of our database for the enzymes that is homologous to THCA synthase or CBD synthase, or CBDA-like enzymes, and we've analyzed it and divided it into clades. So as you can see, there's the CBD clade, the THC clade, and the enzymes that belong to these clades probably synthesize CBD and THC respectively. In between, there's the CBC synthase, and there are entirely different clades out here and even out here, which are enzymes that probably do something else in this pathway. They are functional. They're also correlated with transcript, because the database is also supported with transcript data curated from 4 sources. So we can tell that there are new versions and new proprietary versions of these enzymes, and we don't know their function. So it's up to the synthetic biology companies to actually look at the functionality and the output of these novel enzymes, as an example. And the second example, as I mentioned, in genome editing or gene editing, there are 3 things you need to do for a successful gene editing project. You need to first accurately identify your target. If you have a collapsed assembly, like some of the publicly available genomes, the orange arrow and the green arrow may be either missing, so you have either the green or the orange, or merged into one wrong version, which means you don't have information. And if you want to edit this blue spot here, this particular amino acid, then you need to know what exactly you're editing. So first you need to carefully figure out your target. Then you need to figure out what looks like your target, which is the off targets. and make sure how many copies you have of it, so you actually have, you know how many edits you really need to do. And if one edit is good enough to do what you want, or actually will damage multiple other genes that do other things. And then once the process is done, you need to validate your edits. So you need to edit to sequence and make sure that you've edited the right locations. So you need to design primers that actually enable you to sequence the right edit targets and the off targets. So you need to know what you've done. So all of these are requirements for successful genome editing. And all of these could be, you could utilize our Energene database and have accurate information on the genome content. So to sum up, whether your needs are conventional breeding, we have Quick Genetics with an entire breeding package, all the way from defining traits to actually markers and helping with the production of either stabilized seed or clones. So we aid in the process and make it faster and more efficient of conventional breeding. And if you're into genome editing, either in the plant or in synthetic biology, we have a database called Energene that allows you to drill down into actual gene content and make your edit accurate and efficient. With that, I'll end the webinar and we'll take some questions for myself and for Yannik. Thank you.
Thank you, Uri. I believe we do have some time to take a few questions. And the first one, I think this is for Yannik. And the first one goes like that. Are varieties developed in Switzerland suitable for North America?
Okay. There's a few considerations one has to make. Some of the pain points mostly are the mold resistance of varieties. Swiss climate is extremely humid and subpar for cannabis cultivation. So if a variety does well here, it would also do well in North America. And then there's the consideration of day length. So at what duration of daylight does a plant go into flowering? And for this, we have simulated climates and we do indoor breeding. So I can say that if a plant has been bred from us in Switzerland, then yes, and we have released it in North America, then yes, it performs there. Additionally, however, we've done some field trials and compared common US varieties such as T1, Wife, and the like to Swiss varieties. And we've seen that the yields that were reported from the US are very comparable with what's happening in Switzerland. And also Swiss varieties flower, or the flower initiation of those varieties, is very similar in the US as in Switzerland. There's some locations in the US where one should consider what to do, but even with US varieties, which is in the very southern parts, such as some Texas or Florida, there's some variation to be considered because plants flower much earlier due to the short days. But apart from that, the answer in short is a yes.
Okay, great. Then the next one will be, why did you go into genomics and not settle for selections? Are there traits that could not be integrated using selections? I believe this was for Yannik as well.
Okay. Assuming selection means classic selection without the support of genomic tools. In brief, as long as somebody doesn't do genome editing, he does selection and then has assisting tools such as what Uri has presented or what we are using to improve the selections and the pace thereof. So it boils down to the precision of parental choice and the speed in which a variety can be bred. Most traits are possible with selection, classical selection. However, it's a question of the effort. To give an example, for some very complex trait, it might be necessary to grow out 100,000 plants or even 1 million plants. With molecular breeding, already at the seedling stage, a breeder can determine the most interesting plants to continue his breeding process. Whereas with classical selection, one would need to grow hundreds of acres just to find this one single plant. So if one doesn't do molecular breeding, and there is lots of complex traits in a plant like cannabis, then one approaches impossibility just to achieve the results within time. And this is also one of the differences from cannabis and hemp to other crops, row crops. Wheat always has the same, needs to meet very similar conditions. It needs to be high starch, high protein. Whereas hemp or cannabis, there's so many desirable traits that it's not just main breeding targets, but it's dozens of them. And that's why molecular breeding, again, facilitates somehow capturing this complexity.
Thank you, Yannik. I think the next question is for Wuri, and it goes like that. What are the most suitable traits for using genomic tools instead of classic phenotypic selection?
So this kind of relates to what Yannik just answered. When the traits are dominant and easy to select for by phenotype and phenohunting, you don't need us. People have done very well by classic selection. When the traits are recessive, meaning they alternate generations or need combination of things of certain genes to occur, then these traits cannot be bred for straightforward by classic selection. It's very hard to do. You move one step forward, 2 steps back. So for recessive traits, very complex traits, or stacking of multiple traits, like Yannik mentioned, in cannabis, you need to control flowering time, productivity, powdery mildew resistance, and so on, all in one plant. So if you did that by classic selection, it would take you decades. I've shown in my previous webinar that skunk number 1 reached very nice results for indoor recreational use, but it took 11 years to do so. People in this industry do not want to wait 11 years for a new variety, so you have to use genomic tools.
Okay, and the next question is, how do you organize your genomic data and use it for data mining? Yannik, I think this goes to you.
Pretty sure Ori could give a very good answer too. But yes, absolutely. So it's I'd say 2 things how one organizes the data. It's with a genome browser such as an IGV or JBrowse or what you guys offer. And then for more complex projects, there's always the need for a bioinformatician. So those are the 2 things.
Yes, I could add to that. I don't know if I'd mentioned the Energene database that I described. We could also make a private customized versions for clients with their own data integrated. And part of what we offer with this package is bioinformatics support.
Okay, and I think the last question is connected to that. Can your system help us find multiple gene copies for editing?
So I'd mentioned that in my Energene part. Yes, so we can analyze either the genomes in the database already for copy number. We have done this analysis for specific genes of interest. We could do that for any gene of interest, look at the multiple copies and alleles and variants within the genomes in the database. And we could do this for a private version, incorporating your data or transcript data and trying to find copies and copy numbers of this particular gene of interest in the existing database? So the answer is yes.
Okay, great. Thank you, Uri. Thank you, Yannick. I'm sorry we couldn't answer all your questions, but please know that we will follow up by email to answer all of them after the webinar. Give us a couple days. I'd like to thank Yannick and Uri for their time today and the terrific presentation they shared. This webinar was recorded and we'll have it available in the next few days, so keep an eye for our email with a link to the recording. You'll be also able to find it on our website. If you have further questions or an interesting research project and like to discuss it with our experts, please don't hesitate to contact us at energene.com. Thank you for joining us today. Stay healthy, stay home, and enjoy the rest of your day.